Welcome to the unit on microbial control. In this unit, we're going to look at the various ways that you can control and eliminate the growth of microbes on surfaces and within the human body. We'll look at both physical and chemical means, as well as antimicrobial drugs like antibiotics. As we go through this section, I don't need you guys to memorize what every single chemical does or how every single antibiotic functions. Instead, I want you to focus on why we might need to pick one over another. So what types of things go into the decisions on what type of control mechanism to use, and to understand the overall categories of these control mechanisms and how they differ from one another. The individual details are always things you can look up later if you need them. All right, let's go ahead and get started. In this video, we're going to focus on the physical and control mechanisms for microbial growth. As you've been thinking about how you can control microbes on surfaces and in the human body, it's important to know what level of control is necessary. So for example, do you need to remove all microbes, including endospores, from something, or do you just need to reduce the level of the microbes? In healthcare, that really depends on what an item is going to be used for. So is it critical, which means it's going to be passing through the body into the bloodstream? Those are things like scalpels. Those have to be completely sterilized. So sterilization means we've removed all living cells, all viruses, all endospores, anything that might make a microbe is destroyed and gone. So there are no microbes remaining on the items. That's important because if it's going into the bloodstream, we want to make sure we're not transferring anything that might grow in that bloodstream because your bloodstream should be sterile. If items are semi-critical, it means they're entering the body, but they're not actually breaking through the epithelial layers. So those are things like endoscopes or colonoscopes that might go into the intestine, but not actually break through into the bloodstream. Semi-critical things are typically high-level disinfected, which means we've killed and removed most of the microbes, but the endospores might still be there and there might still be a couple of microbes around. So they don't have to be sterile to enter those types of areas. And then non-critical items, those are things like sheets and pillowcases or anything that's just going on the surface or touching the surface of the body. Those typically we don't worry very much about being sterilized or disinfected. Difference between disinfection and antiseptic is very similar, except antiseptics are used on living tissue, whereas disinfectants tend to be used on surfaces. And this is kind of terminology you probably recognize, especially with all of COVID-19 going on. We've been talking a lot about removal of microbes from surfaces. So something that will remove something on a non-living surface is disinfection. Something that might remove or destroy bacteria on, say, your skin is called an antiseptic. The last level for terminology is called sanitation. This is similar to disinfection, but there's actual guidelines. So for example, if you work in a hospital or you work in a kitchen, things have to be sanitized to a set specification by, say, the FDA. If you're just disinfecting something in your home, there's nothing that says you have to remove a certain percentage of microbes for that to be officially disinfected. This table just provides an easy way to study for those terms. Okay, so it gives you kind of a definition, common ways that these things like disinfection or sterilization are used, and what are some of the main ways that we actually manage to achieve those levels of microbial removal. As we look at different methods to control growth, it's important to note that not all control mechanisms kill microbes. There's some that just stop their growth and others that just remove them from whatever it is that we need to be cleaned off. So control methods that do kill organisms are called cidal, while others that just stop their growth are called static. If we want to measure the effectiveness of various cidal agents, we need to look at microbial death curves. It's important to remember or to think about not all microbes are going to die immediately when you put a chemical or you go to try to control them. Okay, so for example, if you take the Clorox wipe and you wipe it along the desk, you haven't automatically killed every microbe that was on there. In fact, microbes die similarly to how they grow, with an exponential death. A fixed percentage of them are going to die over every time frame. And that time frame is called the d-value. And so the d-value is the amount of time it takes to reduce the population by 90%, which means 1 order of magnitude. And we measure this with a logarithmic scale, which is the red line. And so we can see to go from, say, 10 billion bacteria to 1 billion bacteria, which is 1 order of magnitude or 90%, it took us from this time point to this time point, which was 5 minutes. And so the d-value of whatever this control mechanism is with this particular species is 5 minutes. If we actually started with 10 trillion organisms and we used this control mechanism, it would actually take about 50 minutes to reduce our population down to 0. That means that control mechanism would need to be reapplied and continuing to be in contact with them in order for us to actually drop the numbers. The effectiveness of these control mechanisms then depends on the size of the microbial population. The less microbes there, the faster you'll be able to remove them, but also the composition. So this curve that we have is specific to a certain species. If we use this same control mechanism, but a different species, we might see a curve for a d-value that looks like this. In that case, it looks like we're going from one to another and about half the time, so about 2.5 minute d-value. This means that control mechanisms need to be looked at for the species that you're trying to remove. The other things that are important are the length of the exposure, so how long are you taking to control them, and the concentration of the agent you're using. More concentrated or less concentrated chemicals might affect things differently. You have to consider the susceptibility of the agent to the disinfecting protocol. So for example, something like ethanol that we tend to use a lot for removing and killing organisms doesn't work on the bacteria that causes TB. So if you're working with TB in a research lab, you can't wipe down your bench with ethanol and expect it to do anything because the bacteria are able to survive that. So you have to understand the susceptibility of your organism to whatever you're trying to use. And then you also need to have contact between the agent, so whatever you're using to control, and the target cell. If they're not physically in contact, then whatever you're using can't kill the cell itself. This is really important in healthcare. It's why we have to wash items before we actually go to sterilize or disinfect them, because if you don't wash them and they've got organic material, so dried blood or maybe tissue on there, that blood is going to get in the way in between the disinfectant and the bacteria aren't going to be killed. We also see this with biofilms, so it's very difficult to destroy biofilms and control them because the EPS of the biofilm prevents the contact that we need between the control mechanism and the actual cells themselves. There are a number of physical methods that we can use to control microbes, some of which are bactericidal and will kill them, others which are bacteriostatic and will not. So heat, for example, is one that is bactericidal. You can kill bacteria using heat. If you're going to use heat, the best heat source to use is what we call moist heat, which is typically through either boiling or using an autoclave. Autoclaves are how we sterilize a lot of equipment in research labs and also in hospitals. You can see an autoclave here, and mainly what it is a gigantic pressure cooker. And so what happens is in the autoclave, the pressure is going to increase, and then we're also going to have temperature increase and make steam. The increase of pressure is to make sure things don't boil over, and the combination of increased pressure and steam kill anything in there. Autoclaves are sterilizing, and so they will also destroy endospores. This is different than pasteurization, which also uses heat, but it uses a lower amount of heat. And so pasteurization does not sterilize, it just reduces the number of microbes that might spoil a substance. We often see this in milk. So you don't want to put milk in an autoclave and heat it way up. You'll also denature the proteins in the milk, which means you'll curdle it, and so it won't be milk anymore. Instead, we use pasteurization, so we use lower amounts of heat. There are still microbes in there, which is why if you leave your milk sitting on the counter at room temperature, microbes will grow and it will spoil. But there's less of them in there, so it has a longer shelf life, and we've also removed a lot of pathogens, so you don't get sick from drinking the milk itself. Another bactericidal agent is radiation. So you can use things like X-rays or gamma radiation or UV light that will damage DNA and will kill microbes that are in a substance. We can also use drying, which has been used for numerous years, well before we knew what was actually happening with microbes. For example, if you compare the difference between a steak and beef jerky, you should recognize that the steak needs to be refrigerated, whereas beef jerky is sold in a bag on the counter. It's because the meat has been dehydrated and dried, and without any water content, microbes can't grow in it. And if you pull the water out, all living things have water. We need water to be able to have metabolism so our enzymes can work. And so by removing the water and decreasing the water activity, you prevent microbial growth on those substances. The last one for physical methods on this list is cold. Remember, cold temperatures are mainly bacteriostatic. So they don't necessarily kill microbes, but they inhibit metabolism and they'll slow their growth. So you can control their levels by preventing them from growing, but if that temperature goes back up, they will start growing again. Another physical way to control microbes is through filtration. But filtration is a little different because it's not killing or stopping the growth of microbes. Instead, it's physically removing them from the substance. Filtration can be used for both air, and we see this a lot with HEPA filters, or liquids. And the removal of the microbes is completely dependent on the pore size of the filter itself. So if you use filters that are 0.2 microns or smaller, we typically say that those are going to sterilize whatever you're filtering through, because only the very smallest viruses might be able to fit through a 0.2 micron hole. If you use a filter that's 0.45 microns, typically we say that this will remove bacteria, but it's not going to sterilize because most of your viruses would be able to fit through those pores and would remain in the liquid or the air after it passed through the filter. Filtration is important for a lot of the drugs that we use because anything that has a protein in it can't be autoclaved to sterilize it. The heat from the autoclave would denature the protein, which means the drug would no longer be effective. This is the case for things like insulin or for things like antibiotics that are functioning enzymes or proteins. But if you're going to inject it into the bloodstream, you need that liquid with the drug in it to be sterile. And so oftentimes we will filter sterilize those, passing it through a 0.2 micron filter to remove any pathogens, but ensuring that the protein that makes up the drug itself stays intact and isn't destroyed in the process. There are a wide variety of chemicals that we can use, both as disinfectants for non-living surfaces and as antiseptics on living surfaces to kill microbes or control growth. However, if you're going to choose to use a chemical method, you have to consider things such as the corrosiveness of the chemical itself. This is the reason why bleach is a disinfectant. We might use it on a surface, but you typically don't want to dump bleach on your skin because it will actually damage your tissue also. You need to think about the stability, so how long does the chemical stay there? How safe is it to work with? How easy is it to work with? And what are your target organisms? You don't need to know every chemical method of controlling growth, and you don't need to understand a lot of the differences between them, but I just wanted to mention a couple of the ones that are pretty common that you've probably heard of or seen either in clinics or in your household. The first are phenolics. So phenol was actually the very first disinfectant that was ever used. It was discovered in 1867 by Joseph Lister, and he used it actually on wounds to help with surgeries and to help prevent wound infections. These days, we tend to see phenolic compounds still in things like Lysol, triclosan, which is found in a lot of disinfectant soaps, and Listerine. Other chemical methods you might have heard of are the halogens, things like iodine and chlorine, and these are different. Iodine is an antiseptic. We typically use it on the skin before, say, doing surgeries, or chlorine, which we use all the time as chlorine bleach on surfaces. Alcohols are also antiseptics or disinfectants. You can use alcohol on your skin, although it's drying. You can also use it on surfaces. There's a couple different types. Ethyl alcohol tends to be one that's more common, but isopropyl alcohol can also be used. And then there's surfactants, which are really just soaps. Those are not going to kill microbes, but they are going to remove them from the surface. They help them stop sticking to the skin so that you can wash them away. One thing I want you to notice in the table is that a lot of these, such as your phenolics and your halogens, are at intermediate level, so they aren't doing anything for spores. These are not sterilizing agents. Your alcohols will handle vegetative cells, fungi, and some viruses. Alcohols will actually dissolve lipid membranes, and so they'll work well against enveloped viruses. It's why you can use hand sanitizer against COVID-19 because it's an enveloped virus. But the alcohols typically don't do anything for naked viruses without an envelope. So that's why you have a plus minus here for viruses. But they don't do anything for mycobacteria, and they don't do anything for spores, so these are not sterilizing agents. You do have some chemicals, things like ethylene oxide or formaldehydes, that you can use for high-level disinfections because they can come close to sterilizing as they remove everything, including the endospores, from a substance.